forward. All right. So we are talking about the cells. Chapter 4 is a continuation of the cells, but in a different aspect, from the different perspective. We already talked about all the structures of the cells. Now we're going to talk about two different things here. How things move in and out of the cells, and how the cells make its own energy, which is called cellular respiration. This is just a quick reminder. In order to talk about the movement, you should remember that we have this cell membrane outside, and I'm assuming that you guys already know the details, including the double layer, its phospholipid, why is it phospholipid, and this is the main structure, the proteins, and the protein is the main function. The main structure is the double layer of phospholipid. The main function is the protein. We also have cholesterol and some carbohydrates and so on. So if you guys study, you should know that already. So I already said that this membrane is selectively permeable. It's not freely permeable. It's not impermeable. What's impermeable? IM and AM, all these prefixes means no. No. IM is a prefix that means no. So impermeable, IM, UN, AN, all these means no. So impermeable, no permeability. You don't have a permission to go. Permeability, impermeable. It is not impermeable. How about freely permeable? Anything can go in and out? Still no. It is selectively permeable. There are things, selective things, that have the permission to go in and out. The membrane chooses what goes in, what goes out. We also talked about the DNA. We said it's in the nucleus mainly, but there is some DNA in the mitochondria. We said the cytoplasm, which is this space right here, including cytosol, which is the fluid part, and including the organelles. Now, how can things move crossing the wall? Not the wall, I'm sorry, the membrane. It's different. It's a membrane. Well, it depends on what are you talking about. Not everything is the same. What are you talking about? Are you talking about something like oxygen, carbon dioxide, and non-polar molecules? Do you guys know what non-polar means versus polar? We talked about this before. Does anybody know what's polar and non-polar?
I think it's like for non-polar, they're repelled from one another and they don't have like an electromagnetic attraction, I think.
Yes, but it is more of fat versus our water insoluble versus water soluble. Non-polar is water insoluble, referring to the fat. Remember when we talked about the polar and the non-polar? The non-polar is like the fat that's water insoluble, and the polar is water soluble. So non-polar here means fat or fat soluble. Anything that's fat or fat soluble that is small enough, it can go. How about the water? You said the water cannot go because this is fat. Yes, but remember I told you guys that there are channels for water to move in or out. So these things can cross the wall itself. Others, like glucose and other polar water-soluble molecules, they cannot. They cannot. These are small enough and non-polar, so they can go. How about the water? Is the water non-polar? No, I didn't say that. But water is special because there are channels for it. So those can cross the wall freely. There is no details needed here. It just go in and out depending on the concentration gradient. Like oxygen is more outside, freely move inside. That's it. If the carbon dioxide is more inside of the cells, it will just cross out. Do you need anything to do that? No, it's free. Just move in and out, move in and out, depending on where is it higher concentration. If there's more outside, it moves in. If it is more inside, to move out. That's it. Other things cannot do that because they are polar. And also, they can be large. It's not small like those, and it's not non-polar like those. Anything else, and I told you guys that to make it simple, water soluble and water insoluble, which is hydrophilic, hydrophobic. Just know, you don't have to memorize. Just know one thing. Lipids or fat is water insoluble or hydrophobic. You don't have to memorize anything else. Anything else is hydrophilic. That's it. Just the fats and things related to the fats. So now, how about something like glucose or something that's large, or molecules that are water soluble or ions? They cannot cross. Only this can cross. Anything else? No. Now, if you wanted to cross the wall, the membrane, I'm sorry, the membrane, we are going to make it into two categories. The first one is the one that happen without using ATP. You don't have to use energy. And because you don't have to use energy, we call it passive. So here are the processes, the different ways with which different things can move in and out, can cross the cell membrane. How does it move? Well, number one of the classification, are you using energy or not? If you're not using energy, I call it passive. If you are using, it's called active. So passive, that doesn't use energy in the form of ATP, adenosine triphosphate, can be simple diffusion, osmosis, facilitated diffusion. I know you don't know yet. We will discuss it. I'm just giving you the main points. It can be active. What's active? Using energy, which is ATP. And here we have active transport, endocytosis, and exocytosis. Okay, let's start with this one. Passive. The first one, passive, doesn't need energy. The first one is diffusion. When you say something is diffusing, meaning its particles will have spontaneous movement that's going to diffuse within, like, the water, right? Like if you put sugar in the water, or like in this picture, if you put this tea bag in the hot water and leave it for some time, if you just put it, you cannot drink the tea because you're drinking the water, and just a little bit of the tea flavor itself. But if you leave it for some time, it will start to diffuse. Like here, look at this, how it's concentrated, and then start to diffuse until it will become homogeneous, and now you can drink your tea. So what happened here is these diffused from the inside, crossed this bag, and go out and start to diffuse until it is equally distributed or equally diffused, or become homogeneous. So now you can drink your tea. It is the water is not water anymore. It's all the same. You're not like drinking a little bit. It tastes different than what you drink here. It's the same thing. And this is the point when diffusion will stop. So it leave, and it's leaving from high concentration inside the bag, as you see here, to low concentration, which is the water, right? So the flavor of the tea and the tea itself, it's going to cross, leaving and go out to the water, out. The question is, why out? Because it's more concentrated, as you see here. This is the actual tea. It's more concentrated inside of the tea bag than outside. So rule number one, diffusion means spontaneous movement, but most importantly, what? from high to low concentration. This is the key word. Diffusion, high to low concentration. Very important. Why is that very important? Because this does not need energy. Just move from, you didn't do anything. Just put the tea bag. It will, without any energy, it will diffuse out from high to low. So from high to low, we call that concentration gradient. There is a gradient here. Look at this. This is a lot bigger than this. There is a concentration gradient, meaning there is a difference in the concentration from inside and outside. Inside is obviously bigger. So does diffusion means you move from high to low or from low to high? High to low. So what do you call it? You move up, concentration gradient, or you move down? Of course, you move down because you move from high concentration to low concentration. We'll call that moving down concentration gradient. Since this is moving down concentration gradient, is it active or passive? It's passive. What does it mean? We didn't use energy. So how are you moving? Diffusion via or across concentration gradient. You're moving down concentration gradient, down, which is the difference between the two sides. And one more information to know here is the more concentration gradient, the faster the movement. Meaning, look at here. If this tea bag was more concentrated than that, the particles will leave faster because this is 10 to 0, let's say, and then part of it is moving. Yes, it's 9 to 1. The ratio, the concentration gradient, 9 to 1, and more particles are moving out, diffusing in the water. Yes, it's 8 to 2, and so on. So the bigger the difference, we call the bigger the concentration gradient, the faster the movement.
I say you're under arrest. For what?
For conducting illegal activities at your establishment.
Okay. Special types of diffusion. Special types of diffusion. Diffusion is the big category that's passive. Translation, moving from high to low concentration. Special types. Number one, osmosis. Osmosis is a unique word for water across semi-permeable membrane. If you forgot this, you won't be able to answer your question. Very important. What's osmosis? Water, movement of water specifically across semi-permeable membrane, and it actually moved from hypotonic solution to hypertonic solution. What does that mean? Hypotonic, low concentration.
All of the acids are being
Hypertonic means high concentration. Let me make a practical example. If I have,
high.
if somebody's talking, please mute yourself. So let me give you a practical example. I'm getting a container, and this container, I'm gonna put like a membrane in the middle, dividing it into two parts. One part I will put water, and the other part I will put juice. Juice is more concentrated. We call it hypertonic than the water. So the water will move from the water to the juice. Next example. I will put in one compartment diluted juice, and in the other side, concentrated juice or honey. How the water move? The water move from the diluted, which is called hypotonic juice, to the other one, to the other side, which is the concentrated juice. Okay, if you have two types of juice, you know that the juice, a lot of times they make it from concentrate, right? So one side is having the juice, which is diluted, that we drink, and the other side is having the concentrated. How the water move from the juice that's diluted that we drink to the one that's concentrated. Yes, it is unique, and this is how it works. So look at this compartment. Yes, go ahead.
So it doesn't have anything to do with mass, because it would seem like the concentrated juice would be heavier, which would mean it would weigh more on the other side. So it has nothing to do with the weight.
No, no, it's not the weight.
Okay.
It's just a concentration gradient, hypotonic to hypertonic. So look at this. Look at this. This is, I got like water. I put a membrane in the middle. And this one side, I put the concentrated juice. And then the other side, the diluted juice. Look what happened here. It moves from the diluted to the concentrated. Yes. And this is what you need to know about osmosis. Water move across the semi-permeable membrane from hypotonic or low concentrated to hypertonic, which is higher concentration. You have to know it this way. Now, there are certain terms that we need to understand. If I give you juice, what is the juice? The juice is concentrate. We put it in the water. The concentrate itself is called the solute. The water is called the solvent. Together, it's called solution. One more example. I get a cup of water. I put some sugar in it. I dissolve it. I make sugary water. Or I get one of these Minute Maid powder. I get cup of water. I put a couple of spoons from the Minute Maid powder into the water and I dissolve it. What do you call each one of those? The Minute Maid powder is called solute. The water, solvent. After you put it and you make lemonade or you make the juice that you want, we call that solution. This is one. Number two, tonicity is the concentration. So if I have two things and both of them have the same concentration, I call that isotonic. What's iso? Equal. Iso is a prefix that means equal. Tonic means concentration. Compartment A, compartment B, both of them have this exactly same thing, exact same thing. In both sides, like returning back to this one here. What if I put in this side juice, part of my bottle of juice, I put half of it here and half of it here. Same juice, same one. It's isotonic. There is no difference. But in our example, in this compartment, in this side, I put the diluted juice. And in the other side, I put the concentrated juice. So now the diluted juice is called hypotonic as compared to the other one, which is called hypertonic. Did we understand these terms? Isotonic, hypotonic, hypertonic. If both are equal concentration, isotonic. Hypo means less. Hyper means more. Translation, translation, translation. I'm giving you prefixes and suffixes. You used the suffix tonic 3 times. Isotonic, hypotonic, hypertonic. Tonic is concentration. And then you change the prefix, iso, hypo, hyper. Same, less, more. Practical example. If I get one red blood cell that contains fluid inside, and I put it in a solution that's isotonic, meaning I put it in a test tube that contains solution that's isotonic. What does it mean? Meaning that the concentration of the fluid in the test tube is equal to the concentration inside the red blood cells, which is isotonic, same tone. What's going to happen? Absolutely nothing. Water is not going to move. Scenario number one. What if I put this? Okay, I'm sorry, I got disconnected. I don't know if it's I think it's the internet get disrupted. So let me share it again. Okay, so let's continue. So here we were talking about this. if you put a red blood cell inside a solution that has the same, exactly same concentration, I put it in the isotonic. What will happen to this cell? Nothing. Nothing is going to happen. What if I put it in hypertonic solution? What do you mean hypertonic? Means higher concentration if compared to the inside of the red blood cells. So we compare the solution to the inside of the red blood cells. Hypertonic, what's going to happen? I already told you, if you put it in a hypertonic solution, means the inside is hypotonic. So what's going to happen? I told you that the water is going to go from hypo to hyper. So the water is going to leave the cell, and red blood cells will shrink. Looks like this, shrinking. How about if you put it in hypotonic solution, like in a solution that's less concentrated than the red blood cells? Same rule. What's osmosis? Movement of the water from low to high, from hypo to hyper. In this case, the solution is hypo. The water will leave the solution and go inside the cells, inside the red blood cells. Red blood cells are going to swell, and then it become bloated until it bursts. And we call that lysis. And you need to remember this word, lysis. Lysis means, or lyse means to dissolve. Remember the lysosomes that I talked about before? We're using the same prefix. Lyse or lysis means breakdown, burst, rupture, same thing. So in our cells, homeostasis is an extremely important concept that we need to know. And homeostasis, I mentioned it before. I talked about it in, I think it was chapter 2. And I said homeo means homogeneous, stasis means keep. So you should keep the different concentration and osmotic pressures within the normal range to keep our life. So this is for diffusion, simple diffusion. The next one, which is still passive, is called facilitated diffusion. What do you mean? Isn't that diffusion? Yes, it's diffusion. What's diffusion? Diffusion means you move from high concentration to low concentration, from high to low. Do I need energy from when I move to high to low? Of course not. Diffusion is from high to low. When you move from high to low, you don't need energy. It's passive. Why did you call it facilitated? Because in this case, there is something that's going to facilitate. Still doesn't need energy, but there is something to facilitate. In this case, there is a transport protein. Remember the protein in the wall, there are different types. One of them is going to transport. It will open from one side, take it in, and move it in. Still diffusion, but this type is facilitated. Doesn't need energy. And this, in order for this transport protein to facilitate or to assist, it does not help anything. No, it is very specific. Like this one here is for this type only, whatever it is. Let's say this is glucose or something, or whatever it is. It should fit into here, and so act like a key. And this protein, transport protein, is going to facilitate its diffusion inside. What if I got something else like circular in shape? No, it will not fit in. So we mean transport is specific here. You have to fit in. So this is just repeating diffusion and osmosis. Water and other things diffuse into and out of the cells, up or down its concentration gradient. It moves down concentration gradient. Diffusion of the water is called, you guys know? What's the diffusion of water specifically?
Osmosis.
Osmosis, yes. Diffusion, is that active or passive?
Passive.
It's passive because you move down concentration gradient, and because of that, it does not need energy. The mechanisms adjusting all of this is called homeostasis. Facilitated diffusion use transport. What? Transport
Proteins.
protein. Transport protein. Is facilitated diffusion use energy? It doesn't use energy because it's called diffusion. It does not. So here it is. So we talked about the three passive, three active ones, active transport. It's called active, so already called active. You cannot be confused. Plus endocytosis and exocytosis. So active obviously means you need energy. Why do I need energy? Because this time, which is the opposite of what we talked about before, this time you're using against concentration gradient. Look at this. There is lower concentration here and higher concentration here. Remember in the diffusion, we said you move from high to low. Now it's the opposite. You move from low to high. So you are like squeezing yourself in this high concentration. So that's why it is active. What should I remember about active? Against concentration gradient. From low to high, you have to remember this. Diffusion is passive. What does it mean? Doesn't need energy. Why? Because it is diffusion. You move down concentration gradient from high to low. This is active. What does it mean? Using energy. Why do I need energy? Because you're moving against from low to high. The first two here are called endo and exocytosis. Translation. Endo, N. You're moving N. Cyte, cells. So you're moving into the cells, endocytosis. Exo means out. So what's exocytosis? And cyto means cell. Osis means process. So three parts here. Exo, cyte, osis. Exo is out. Cyte is cell. Osis is a process. So exocytosis is the process of moving out of the cells. Use my way, this way, which is the scientific way of cutting into different pieces. It will be a lot easier. So if you're moving something, endocytosis means something is outside and you move it in. Is that active or passive? We're talking about active now. Because this is something that's big enough that you cannot diffuse it. Endocytosis. Now, depending on the nature of what you're moving in, if it is something solid, we call it phagocytosis. What's phago? Cytosis. Phago, eat. It's a prefix that means eat. Cyte, cell. Osis, process. The process of how the cells eat something, meaning it moves in. It's endocytosis of a foreign cell or a substance, something that you wanted to move into the cells. It's phagocytosis. Like your white blood cells, when they find a bacteria or a virus, they can actually eat it. So the bacteria is big enough. You cannot like it cannot move by diffusion, for example. It's not like oxygen or carbon dioxide or stuff like that. It's big. It's a whole organism. So you cannot. The cells will eat it. Endocytosis. And we call it engulfing, eating, engulfing. Exocytosis is the opposite, which is basically something like the paramecium. It's one of the cells. When they get rid of something, push it out of the cells, that's something big, you call it exocytosis. Look at this. Here is, these particles are big, so it cannot move in by diffusion. It cannot. What should I do now? You are going to eat it. Look, it push in, making a vesicle like this, and the vesicle detach in, moving it into the cells. 1, 2, 3. This is endocytosis. Here is the opposite. If there is something inside, you want to get rid of it, merge it to the cell membrane, release it out. Both of those are active transport. And here it is. In active transport, is it protein or lipid pump? We just mentioned it. It's the protein. And you move down or against, you move against, otherwise it will not be active. What provide energy to the cells? ATP, obviously. Exocytosis, is that in or out? It's exo, out. How about endo? Endo, into. And last slide here, and we will do the rest later on, or Thursday. Obviously, from all what we talked about so far and other things as well, we always need ATP, which is the form of storing our energy. You store it in the form of ATP, and when you need it, take the ATP and you get the energy out of it. And here is how it looks like, just to give you a brief idea. You guys were asking, somebody was asking about it. Adenosine triphosphate, meaning adenine, and you need to know this. Adenine is a base. If you add sugar to it, which is called ribosome, now we call it adenosine. What's sin? S-I-N. It means sugar. Sin means sugar or carbohydrate. So adenine plus sugar, adenosine. And then you add 1, 2, 3 phosphates, adenosine triphosphate. I'm going to stop here and stop